Who wants to be? Okay, well, welcome. Maybe, do you want to do with short introductions again, or you feel comfortable, you know, everybody? Maybe.
I was thinking go to nice university.
Yeah.
Jennifer. I'm AJ, also from Rice.
Zach, University of Oregon.
There's a Mike from the UC San Diego.
Okay, wonderful. So, we only have one prompt for this, and, you know, take it away. We would like you to begin, now that you've heard the, you know, outstanding questions and some of the tools that are available, both through the talks and the previous breakout session, we would like you to begin identifying potential connections that could be made between, the questions that you've heard and the approaches, that could connect synthetic biology and aging. These ideas don't need to be associated with your personal areas or anything, but they can be. So, you know, based on what you've heard so far, how do you think these potential connections could be formed? And first, before we start though, we should identify a person who will do the note-taking and the report out for the next session. So it could be anybody, but I was advised that it should not be the moderator, so one of you guys, feel free to flip over. This is notes from the previous session, so we can just flip it over and we can write down the ideas that we discussed, and then whoever writes them, and you can take turns in writing them, we'll report it
This
out.
looks like good notes, sort of what we talked about in the last session.
Yeah.
Almost like a nice starting point.
I'm happy to take notes, but my handwriting is atrocious, so I did the last one on my computer. If everyone's good with
That's what
that.
I did also, I did mine on my computer for the last session, I can't, my handwriting.
Okay,
Nobody
as
was
long
reading.
as you send it to the co-chairs, because they will need to do a synthesis for tomorrow morning, so if you can email it to them, then that's perfectly.
Who should we read them to?
The co-chairs,
Oh,
Josh and
Josh.
Bridget,
okay,
yeah, either one, and they'll exchange it.
okay.
I'm happy to take notes.
Okay, great.
So I guess sort of, jump off thinking about this prompt a little bit more here, the connections and research questions that I almost want to start back a little bit before, just characterizing genetic expressions to aging and stuff like that. So if I wanted to potential connections and approaches, one of the things we just don't have a really great grasp on is all of the pathways, like what is every gene doing? What is all the different expression profiles, things like that, that relate to aging, particularly in worms. I know that that's something that's still true. So in terms of building, from the synthetic biology side of things, like how are we going to build these systems to detect or to understand the full expression levels? Something like that would be very helpful across the time that it's aging, right? As it's getting older, what are those expression changes always looking like? And we still, to this day, don't have a good tissue by tissue kind of way of understanding that, or have done that yet, or, say, change the aging in this particular cell type or something like that, or extended a particular cell types, how long it's lived for or something like that in principle, but.
So where do you think, thinking of that big question, where do you think other methods have failed where synthetic biology could be helpful?
Well, other methods that I think that have still done great, but haven't done as well as I hope, comes back to something we were talking about in the last section of single cell sequencing, right? It's really just the tip of the iceberg in terms of being able to understand, where all these transcription factors and those low expression genes are. But one of the other things that again came out of the last session was like, oh, well, then, there's ways of doing amplifying signal and stuff like that. And we don't have a lot of those in worms, at least very few people have done stuff that's similar to that. And then again, we just do it by tissue and everything as well. So I think that's one of the connections that I'd really like to see happen is like, okay, we can do bulk sequencing, but we haven't really, and we can do some tissue specific bulk sequencing with FACS sorting, something like that, but we really have not gotten to this, really high resolution depth for RNA sequencing across tissues and across ages. So I think if there are, well, I don't know what those sensors actually would be for detecting lower transcription levels, things like that. So if other people have ideas on that, that would be wonderful. And again, even though you're trying to translate these to worms, because that's just the world that my head lives in, but you know, the same concepts can be applied from the cell culture world and then the cell culture world and Easter bacteria, and then just bringing like, oh, we'll use these kind of synthetic promoters that, you know, there should be some sort of, you know, orthologous connection point that.
So sensors for detecting low level transcripts in a something specific manner.
Yeah.
Okay, do you have
That's great.
anything that as a symbologist?
So this would be something that you would do in single cell but across different tissues.
Mhm.
Because one of the things that we discussed a lot in the other group is how much this is aging is a problem that we can address studying a single cell, and how much is an organismal problem, right? Like when we think about aging and we age and say, we talked a lot about all this evidence that as we age, our ability to exercise, for example, goes down and muscle mass goes down, but it can't just be as simple as all the cells in your muscle, right, stop working and it just becomes senescence. There might be some sort of a system level signaling that is also involved. And so if you think about studying this, then should you have model systems that allow you to study at the interface of different cell types in a way that is more kind of organoid-like type approach? I don't know, because I don't know enough about the biology of aging.
I think that would be great. I think that's something that we talk about in our lab, being one of those like organoids and applying that, because we're realizing, you know, it's also kind of discussion at lunch and stuff like that too, right? Like, we're never gonna do, you know, as we're building all these synthetic networks to study aging, we're never gonna put those in humans, right? So just because of all the ethical barrier and the challenges of that too, so.
No,
You know,
but to
building
study
an
it.
organoid type system, yeah, study it would be great, maybe in this organized fashion, that's stuff that came in our session as well before, and that would be wonderful. To be
I
able
guess
to-
to go back to your idea, which I think is really powerful, to have some sort of high throughput way to study, say, transcriptomics, transcriptomics analysis and look in the aging context, how much it seems that, you know, getting fibroblasts and putting them in a filtration lab and culturing them is not a good model for aging, right? So then if you do that type of analysis, how in single cells, how useful is that going to be, right? Are you gonna get something that gives you some information about aging versus simply fibroblasts in cell culture
Mhm.
growing rapidly?
Yeah, and integrating things like the immune system in there, right? So that's tricky, so it's, you know, it can actively inhibit repair, right, which might be a cause of, you know, sarcopenia with aging, in addition to the metabolism.
Yeah.
That's another thing that came up in ours, is how, you know, ways we can detect them and monitor metabolic metabolites that might be responsible for on a cellular level, but then if you have just a small number of cells starting to do that, how does that spread in the system?
So maybe one of the potential connections would be to use synthetic biology tools to create some meaningful model systems to study aging.
Well, how would they look like?
I don't know, because I don't know enough about aging biology,
Well,
but
I guess
something,
to see something like the transcription factor
Yeah.
changes, right? And then potentially even, you know, can you have transcription factor changes have different effects in young versus old also?
Like, here's one thing that I've been going back on it for you know, I don't know how long, maybe 6 months now, like, we don't have really good RNA editors yet, right? With the CRISPR-Cas systems, there's, you know, we can basically make, you know, small, you know, prime editing, like, changes in the RNA. There's not really a way to do like big tagging or anything, as far as I know, right? We don't have any way to like add RNA sequences to RNA in vivo, right?
RNA to RNA.
Mhm. I don't think that exists yet. So if that's not something that we actually have that we can't do yet, well, I mean, you could imagine a synthetic bio approach where you're, you know, tagging, you know, you're targeting this, you know, you have 100 genes you want to target, they're all potentially low transcripts, right? But if you could target it with something that's a, you know, you could then do like a biotinylation or something, I don't know, or some sort of RNA tag that you could then use to do that pull out with. Later on, you could actually do some sort of quantitative measurement of that particular RNA sequence. I'm not sure how that would work, but maybe something like a, you know, something fused to a T4 RNA polymerase or something like that would be a way to like tag it into there, I don't know. That's where I want to get like a protein engineer in a room in an RNA biologist to be like, okay, so if you were going to actually add additional sequences onto a native mRNA, how would you do it? And then see what they would say, like, those would be something that I would love to just, because then, yeah, you could do all sorts of things with like, oh, I have these 400 genes, let's see what they're expressing.
So how to modify native RNAs in a high throughput manner?
Yeah,
In vivo.
in vivo.
Can you, okay, so it has to be active. It can't be a destructive process.
I would imagine, well, I don't know, I guess I'd have to see. In a destructive, I mean destroys the RNA
And destroys the cell,
and destroys the cell.
meaning like you can take an endpoint measurement versus like a continuous in vivo measurement.
I think you could destroy the cell, I think that would be okay, I mean, again, I always just mentally resort back to forms, because that's my comfort zone on this, but it's like, you know, they're all clonal, because they're self-relenting hermaphrodites, so if you just had a population that was genetically identical, and then you age them to a certain time point, I was like, okay, I wanna, you know, induce my system, I'm gonna tag all these RNAs now, and then produce, you know, pull them out, and then it's destructive, which is fine, but then it's also of the same cohort, genetically identical and of the same age as a bunch of others, so that hopefully it's still representative of the genetic state, the age state, the metabolic state of that same individual.
If I'm understanding you correctly, then you want counts of the RNA, for example, the counts of 400 transcripts relative to other systems, but always the same RNA sequences that you're interested in probing?
That would just be one potential way of doing the system, right? Because like we have, you know, again, having the challenge of sequencing low transcript RNA and getting a nice quantitative response to that. So yes, I think that's one way of thinking about it, is saying, okay, we have these 400, you know, genes. I want to know how many transcripts they have. And maybe the easiest way to do that would be to have a universal tagging modifying system to put something onto that. You'd always pull it all the way out, so you're not biasing yourself to pulling out specific, you know, lengths or whatever. You're always pulling out the same thing. I don't even know if that's possible.
How many genetic modifications can you make at once in a cell? Or like,
For
could
a
you,
worm,
yeah, could
I
you
mean-
feasibly add like a barcode to the end of all of those genes that you're interested in the base type?
in the DNA level,
Mhm.
in principle, you could add several hundred, yeah.
Okay.
I think that would be challenging. We've never done it.
At that scale, yeah.
But in principle, I think we could, right? So that's what my work has been on, is like, how do we do the high throughput genetic engineering, right? And we've been focusing on how do we do, you know, one site, but thousands of different versions at one site. But the same kind of logic we could use by inducing, you know, create this library that has homology donors, just like standard CRISPR, right? Homology that insert the sites. There's nothing that prevents us from being able to tag multiple sites all at once. As well, and just having that library of donors having homology to unique sites, having the RNA sequences present for it. So I think we could do endogenous site editing at a large scale, but then, you know, it's how much of that transcript. I mean, I guess you could then use something that you're editing at the DNA level, and it's gonna come into the transcript there. So I guess that would be a way around it.
We routinely use like unique molecular identifiers and barcodes that are tagged to the end between the stop codon and the poly A tail that we're introducing, like a 10 degenerate base pair
Oh, really? We're
sequence.
doing that.
So how do you do
Okay.
that?
We do that when we're putting them in as transgenes. So we
Okay.
tag this sequence on the end of the genomic or the end of the DNA, and then we introduce the transgene.
So you have a barcode instead of like the reporter? Like,
We have a barcoded reporter.
So you barcoded the native gene, and so that becomes your unique identifier, so you do all the demultiplex, the deduplication later on a bulk seq or something.
Essentially, where we can, and then so you can read out the protein level via fluorescence, whatever, fax or whatever.
yeah, but now that we have issues with, or the recognition of, you know, the issues with the GFP reporters and things like that, activating the immune system,
Sure,
it'd be really nice to have just a simple barcode that you can figure out your sequencing.
yeah, and then we only amplify the barcode with PCR instead of wasting reads on the whole transcriptome. And then we can read out and deconvolve how many counts of each of that barcode are present in the population.
So the
That'd be really
barcode,
cool to like monitor some of these rare cell types.
is the barcode gene specific then?
So in our case, we're putting it on a specific gene. Yes, we're tagging it, for example, to the end of a fluorescent reporter or whatever reporter you would choose. And then we're introducing the whole genetic cassette at once
Okay,
into the system. We're not actively tagging a native gene. But with prime editing, you can do those short sequence introductions, which I've never done. I have no idea how hard it is. But if you did a multiplex prime editing and put in a degenerate barcode or a known barcode, you could then pull that back out at the end.
so in that case, you would, okay, so if you want to do a lot, I could see how that could totally work then. So if you just did this in parallel, right, you just did 100 different genes, because cell culture work too, you just do one cell at a time or something, like that, right? So could do that in worms as well, where you just had a bunch of individuals, where you had 100 different plates, and they all represented 100 different genes, where you barcoded them all individually, and then measured the state of that particular low transcript. I feel like that should have, has that not been done though? I don't know, I feel like that maybe would be the way to.
Write that down.
Yeah. Additionally, though, you could, if you know the sequence of the gene that you want, you could use primer amplification of that specific gene
Yeah,
rather
we can do
than
that,
tagging it.
Yeah,
And then you don't have to do any modifications on the cells. You can use the wild type cell and pull out the barcode at the back, add the barcode at the back end.
that's true, yeah, because that's if you're focusing on just one, too, right? So,
Mhm. Which, I mean, if you multiplex your primers, depending on how much homology there are, you theoretically could, multiplex up to whatever. That's essentially what the cancer companies are doing for when they're doing like the whole exome or the targeted exome sequencing. That's essentially the platform that they're using is
Yeah.
they spike in primers during their Illumina run to only amplify certain sequences instead of doing the whole transcriptome.
Yeah. I think the power of the multiplexing approaches though too would help for say, if you want to do genetic perturbations or metabolic perturbations or different environmental perturbations, rather than having one different tag gene at a time, right? Where you have to do all those perturbations across 100 lines or 400 lines or 1,000
Right.
lines. If we had this, I'm just gonna call it this magical RNA protein CRISPR, where you were tagging something on at the RNA level or doing it at the DNA level where you're actually barcoding things in, 1,000 at a time. Then you actually have the throughput that's within, an R01 budget to actually do these kind of experiments where you're, could say, okay, I have my one strain now or my one cell line or one thing, and I want to do 100 or 10, 1,000 perturbations, whatever they are, and then measure these, 1,000 different low transcripts. I think that's where the power of a high throughput multiplexing approach would really come in.
Mhm.
That's what I would like.
But
So what AJ said could potentially solve that problem you just raised. Do you think that's a feasible approach?
I do, yeah, I think it's, yeah. Well, doing it at a single point or in a multiplexed, so where there's a lot of different.
It's up to you,
Yeah,
it's your project.
Well, I think, doing the barcode component of it, I think that would be fantastic. Doing it in multiplex fashion, I think is where the synthetic biology challenge comes in, right? Because if I think it's simple nowadays to do one genetic edit and putting in one, small barcode for any particular sort of gene. But if you wanted to do, 1,000 at a time and have that cell line or have that worm or, mouse model or something like that, that becomes a lot more challenging to do, just because of the scale of that. So that's the part that doesn't exist yet, but the barcode itself, that's actually a great, fantastic way to actually measure the quantitative amount of it, because then you just do the deduplication after sequencing, right? So you do the bulk essentially, and then each one has a unique molecular identifier via the PCR component. So that should work.
And you can go
Yep.
clonal with the worms, right?
Yep.
Which makes this possible
Mhm.
in other cell types where you don't have enough time to go clonal or you can't get them selected down to a single cell.
Yeah.
The system doesn't work very well because then you would have heterogeneous molecular identifiers for each gene that you're interested in.
That's true, yeah.
But if you did a degenerate barcode that you're like, if you're doing prime editing, the part that you're inserting behind the genes is degenerate. And then the targeted sequences, you have a library of targeting sequences. Then every insertion would be unique. And then you go down to a clonal worm, each one of those integrations would be unique. You can sequence those, figure out what the barcodes are, and then you can figure it out from there.
Yeah,
Without that clonality, it won't work though.
that's true, yeah. No, that is one of its great advantages, we can't go clonal, because the self-relating, they actually can't outcross as hermaphroditically outcross, right? So they do have males and hermaphrodites. They don't have natural females, although we can genetically make them. But yeah, we can force it to be a clonal population entirely.
That's awesome.
Great, what other potential connections, that you could
I think we
see?
wanna get this to mammalian systems, complex,
Okay,
with multiple different cell types. Even if it's a in vitro culture with
Mhm.
multiple cell types in 3D, I think it just is not gonna translate to human aging until we do that.
Right, absolutely. It'd be awesome. If there's a way to do mouse embryonic stem cells and then grow a mouse out of that, right?
Yeah, I mean. Yes, but there's gonna be an easier way.
Right.
I mean, can't we, even if we take, let's say, XYZ cell populations and then create a 3D tissue from that?
Sure.
I mean, so we don't have to go through the organism potentially.
Mhm.
How hard is the transfection? Where you're building it from starting? When you need it, but how are you gonna get the transgene in there?
Mhm. So I use nucleofection for primary MSCs. We have another student that uses electroporation with the neon system he uses mouse embryonic stem cells. One of the students uses lentiviral transduction for her T cells. And then all of our our hex cells and all those other like normal cell lines, we're using chemical transduction or transfection, excuse me. So it really depends on what the cell type is and whether or not you can go clonal. I can't go clonal with my MSCs. And so I have a little bit bigger challenges than what my colleagues that can go clonal with sorting via hex cells can do.
But if you have your MSCs, and you potentially build some type of 3D tissue with MSCs. Right, since you still retain the progenitor quality. And
I do.
differentiation capacity.
So the primary stuff that I'm using the MSCs for is not their stem potential. I'm using it for immunomodulatory and as like a infusible cell type.
Mhm.
There was another student that was using bone marrow MSCs that was still retaining differentiation potential. But him and I have not really crossed
So if
paths
you're
on
gonna
that.
transplant those, could they be, we talked about sensor systems, could they be used as a tissue sensor if you're gonna be transplanting them in vivo?
Yeah, if we could get a good sensor actuator that they could read, absolutely. I mean, even hex cells that are covered in, what is it that Omid uses? I can't remember what biomaterial it is. Yeah. Basically, he encapsulates them in an immune covering, and then he can put them in the body anywhere. Caleb's working on with him to do like sensors for that. Jeff Tabor just did that with bacteria, right? And sensed chronic inflammation via the bacteria that were covered in this
You can
biomaterial.
make different materials that allow you to contain the cells, but they're permeable to.
Yeah, that's the field I used to work in, hydrogels for
Yeah.
encapsulation.
Encapsulation, that's the word I was trying to think of. Thank you.
Yeah, but I'm also just thinking of delivering them and embedding them. You know,
Yeah.
if they go to areas of inflammation, just let them embed.
Right, circulating where they will.
Yeah, and then they're gonna sense, yeah,
It'd be phenomenal.
we see too much enough information around the materials.
Yeah,
So I
I
don't do that
bet that
anymore.
makes sense. What if you were to infuse an MSC and you would want it would go get to inflammation, what would you want it to read out?
So I wanna see some tissue signals that are related to potentially hypoxia, fibrosis markers, maybe some metabolic markers. Things like that.
Hypoxia, metabolic, what was the second one?
Some, yeah, fibrosis
Fibrosis.
and potentially some inflammatory markers.
Okay. And how would you read it? Like what would you be your preferred, sky's the limit, how would you want it to talk to you as the scientist or the physician?
I mean, the easiest thing from the physician perspective is to be able to take blood, right? But that's hard, right? So I'm thinking of your barcode and some sequencing maybe is a primitive first one.
Mhm.
So you have to actually harvest it and look for the readout.
One of the things maybe you'll tell me whether you think this is actually possible. One of the things I've been thinking of is MSCs secrete exosomes, just like everything else does. And you can encode RNA into the exosomes. So then you could have the RNA be traveling throughout the body as a measurement of these different programs, and then read that out in the endpoint. Instead of having to go get them, it'll just be very low abundance.
Yeah. And people are looking at that for cancer-related exosomes. in the blood.
Exactly.
For the synthetic people, like how hard would be to like say engineer a cell and let the cell function as like a part of the circuit to like interact with other cells, to read and act. Is that?
I mean, that's a lot of what you're working on, Laura.
Sorry, I wasn't listening, I was thinking,
Oh.
I'm so sorry.
I'm just wondering like how hard would be to like engineer a cell, but let the cell function as a part of the circuit. Like basically a cell circuit, like you engineer a cell, let it read signal and like act.
Right, yeah, no, I think in terms of like a technical challenge, I think that's something that people are overcoming. Like, there's a lot of tools in terms of both delivering the circuit and like engineering the cells, and the main issue is gonna be stability, but I think that, it doesn't seem to me like that's the long-term challenge. It's something we're addressing. Is more than we just injecting these cells, right? In tissues circulating, re-engineering cells that you're kind of reimplanting. Like, I don't really know what the future of synthetic biology from that perspective is in terms of therapeutic
I'm
applications.
thinking of some, I don't know exactly how this connects, but it just somehow came up in my head that, a collaborator, Sharon Perret, she made vascular networks in the hydrogel. And then she could stiffen the hydrogel, sort of like stiffening with aging, and could then look at what happened to the vessels. So that'd be something where you wouldn't have to make, you wouldn't have to put it in vivo or whatever, but you have some model that recapitulates an aspect of aging, and in this case stiffening, and then you can look at what's going on. If you could have 2 or 3 cell types in there.
So I think in general, there's huge potential for connection between synthetic biology and aging in making model systems, right? Because we can study aging with, we can study with cell culture, we can study just by observing human aging, like the time scales obviously are not.
Yeah, we get old mice, right?
Yeah.
We have aging colonies of old mice with various genes.
But if you start thinking about, for example, doing screening for drugs or things like that, that becomes like really hard.
That's
And
what,
looking at like high throughput and just kind of doing it in a way where you can actually develop therapeutics in a timely fashion.
that's where the worm
Oh,
come in really, really heavily, because at our lab, I don't work on this directly, we have the CITP, the Senior Intervention Testing Program one, where they do just have a bunch of plates
yeah.
full of different drugs, and because the worm only lives like 2 weeks, it helps make it a really great model for doing these large scale drug interventions. But one of the things we talk about a lot, and I don't know, again, this is another thing where we talk about solutions to this one, and it's probably a synthetic biosolution thing too, is that when we're trying to drug worms and do these things, we don't actually know all the tissue types the drug gets into. Because it's not like we can just like feed it a pill and the worm takes in a pill, right? And we know where that goes, right? The worm's just existing on a plate where they're either absorbing it through the cuticle or eating it through the bacteria that's coming in through that. But these drugs are in different states. There's DMSO, water, and things like that, different, how long it's lasting on the plate. All of that depends on the environment, the thing too, right? So, one of the challenges I think from our perspective is when we want to do the drug screen things is how do we know what tissues it's getting into? Even to, right, there are certain drugs that don't work in the worm that do work in other systems. And then it comes down to, okay, well, how did you know you drugged the worm? And so it's like, well, we probably need a sensor or something like that to really bring those in and understand that that's actually happening.
How are kind of things correlate between the worm and mice or people as far as the various mechanisms
That's a great question. I don't know. I do know. Yeah. Trying to think about which.
For interventions.
First, even screening.
Two of the interventions that work in the mouse intervention testing program also extend lifespan in this. Out of 71 compounds tested.
Sorry, well,
71 compounds tested, 2 of them. Lifespan extension,
and the mouse,
both ones.
mouse and the.
The sinusitis has 3 strains of elegans, 3 strains of Breakout, and 3 strains of tropicalis. Tropicalis seems to be refractory almost almost feelings.
I think if I'm remembering, it's like Briggsy and Elegans will respond to a lot of different things, but then Tropicalis is the one that will go, right? So I guess part of the, I don't work in the part for it, but yeah, for whatever reason, Tropicalis is just one that just doesn't respond.
Don't know if the drugs get in, don't know that ones.
Yeah.
A lot of blanks.
Yeah.
But the C elegans gives robust response, but they're also heterogeneous. So if you look at so it's reproducibility across 3 laboratories, that's critical. And all the sources of variation in the outcomes have been identified. You talk to Patrick and deal with filtering on all.
So like the microbiome and the mice.
And that's less clear. They're all fed or things. A lot of it's very extension, like a very good assumption. In sinusitis, most of lifespan seems to be coming through the neuronal system. Probably true in other organisms. So the question is, are we getting a drug? So for interventions, lots of.
Well, also another thing, so we study the dynamics, so we, saying the dynamics can matter both on the cell level and on the tissue level. And then when we talk about dynamics, the timing really, matters. So like if we want some like intervention to really, like reprogramming some cells. And then it looks like ultimately there needs to be like a certain way to get a real-time control of your synthetic system, but is that something that's like a very feasible, I don't know, a real-time control of a synthetic circuit that you can put into a cell or tissue?
That's,
Like
yeah, what
a,
we try to do to improve the dynamic resolution of the response of these circuits.
but then I know we can use inducer, maybe you can use the photogenetics, but if like one day people want to develop a certain treatment that can actually put into the patient, but in that case, is it like a real-time control kind of feasible?
I think more, I mean, most of what the idea of creating a kind of sensor response based on a genetic circuit is that you have the other regulation within
okay,
the cell.
so
I
it's
think
the
the time of response is gonna be kind of critical for that.
Yeah,
If you're creating, say, a diagnostic or some therapeutic that is activated in response to a certain aging marker, the response doesn't take, longer than the aging process, or like it operates over the time scales that are relevant.
But like in the aging context, and maybe the whole environment kind of degrades. Maybe the synthetic circuit, or I mean, although it's designed to have a very precise auto regulation, but in the end, it could get like a very malfunctioning or at least getting very noisy. I'm thinking like maybe in an aging context.
I think that's an issue for any synthetic biology application.
Okay,
right? You're
like a
looking
very
at-
stochastic.
Well, it's not, I don't think it's necessarily that. I think you are having to deal with, metabolic burden, for example. But the whole idea of creating these regulatory systems that operate at the single cell level is that each cell essentially will deal with the noise. Like the regulatory marker system in each cell will address the noise so that you're not regulating the response at a cell population or tissue level, but each cell will regulate the time of response. That's how you address noise.
Also, just out of curiosity, is there any theory about like whether we can like leverage, we can exploit the noise? Like noisy nature of the regulation? I mean, is there any theory like that?
Exploited in what kind of way?
I don't know, I think it's very vague, but like, some certain synthetic circuit that can perform differently under high and low noise. I don't know, like, it's very vague. I mean, like, currently, like, the noise or stochastic in cells are considered as something that can interfere with the synthetic circuit. But is it possible, like, that somehow you can actually use the noise to?
I think, when I think of noise, I just think of perturbating the metabolic state from like a homeostatic, right? So if we wanted to make a stress response or something, right? So if it's noisy, or maybe there's a lot of stress that's happening on the cell or something like that, right? Causing the noise to be broadened. From a synthetic bio standpoint, thinking about the worms, I know we do have, common stressors, heat stress, right? And so we can either have they naturally will live in environments that obviously aren't incubators, right? So we do have heat shock reporters, heat shock expressible transgenes that we can use that will then respond to the changing temperature. So I could imagine if I wanted to develop a circuit or some pathway, just simple genetic engineering, maybe it doesn't even reach the synthetic kind of level from that, right? You could imagine a system where the heat sort of is going up and down quite a bit. You're modulating the expression of something else due to that heat shock stress, which is then also responding to more fluctuations or stochasticness that's happening in that environment, more stress. So that would be, I guess, a synthetic circuit that would then respond to the stochastic noise in a way, at least in temperature.
And Aaron O'Shea create a lot of circuits around noise.
I'm not sure. That sounds familiar, but I'm not as familiar with that myself, sorry.
There's also the Kamash lab, I think. They do a lot of work where they're trying to build true control systems that are robust to noise. And so they have to have a way to sense it and bring it to baseline very quickly, to where it does not oscillate from set unless user defined. So maybe we can see if I can find that paper that just came out or send it to you.
Okay, cool.
The radio systems become noisier. Systems become noisier.
Yeah. Synthetic system definitely would, because we see all the transgene silencing that is being thrown into the system. Breaking the tightly controlled and defined system from set point 0 to set point N. We would definitely see that there.
So you have ways of monitoring noise. If you would, in aging, if you activate or accelerate one of the hallmarks of aging, you might be able to see a readout on how noise levels change. Because one view of aging is that you have, during development, you build up this system, and the robustness of the system comes from the ability of the components to exchange information, that sustains them. But if they lose sufficient synchrony of response, which is an increased noise, and they degenerate, their functions no longer support them. So a reporter of noise is independent of it's really several reporters would actually be able to address. Then you can determine if there's a threshold for the aging phenotypes, because of course you want to really get physiology.
And how do the epigenetic changes that happen with aging affect the noise? And is that part of noise or?
Probably.
I think probably yes. Like the epigenetic reprogram. I think the written paper from David Sinclair, they say like epigenetic reprogram can distribute the factors more evenly. So like every gene expression is kind of more evenly, so there's less information entropy, I suppose. Just like I was saying earlier. So essentially, like everything is more homogeneous, and the cell cannot like maintain its identity. But okay, is that noise? I think maybe it's a certain noise because like essentially a more pattern expression profile is now more even.
Yeah, okay. This, one more example. So this work from host substance on complexity increasing. If you look at quite good patterns, we call multiple points. But there's synchrony that require electrical propagation. So that's an example with noise, still materials. But noise also synchronations.
Yeah, that's
Well, it sounds quite right, but it's not just signal to noise that's classified by chemistry. In the noise distribution.
Makes me thinking about the noise and stuff like that. It's one of those, going back to like a more classical synthetic bio where they built the battery, those the oscillators, right? You can turn on, turn off, stuff like that, like. We have all of those same, fluorescent things in animals and worms too, but we've never built an oscillator. And I think something like that would be super cool to just build up and it's like, oh, let's look at the neuron, let's look at something. It's like, does it blink it couldn't be 18 minutes like in the bacterial one, but maybe it's every few hours we see these things going. And then as aging would go on, you could then say, but again, getting back like, oh, is this tissue, maybe a young tissue flashes every so many hours and then an old tissue flashes 2x that many hours or becomes more stochastic or something like that.
So that's circadian clocks that are both central and peripheral, and they lose their synchrony. Like that.
Yeah, exactly, yeah.
I think it'd be likely that it turns off over time, like overall, the program gets shut down rather than it necessarily changing in dynamic. Does that make sense?
So it keeps the same frequency, but then it stops one day or loses its fluorescent,
Something to that effect, like
Kind of dampens.
yeah,
Really?
to zero.
Yeah, usually
Okay.
that's what happens with the-
We see that in our embryonic stem cells, not mine, but that when my colleague puts in these programs, he'll have, 100% positive when induced. We don't do oscillations, it's more like the toggle on off. So induce it, it turns on, and then a couple passages later, you turn it on and it's only on 50%. Another passage later, it's on 10% and then nothing.
So the oscillation has an on-off switch, but the oscillation does not have noise. Or the oscillation goes on or off, and that's the noise, but there's no noise in the oscillation itself.
I think you can have noise in the oscillation, but the oscillation is generated by a specific pattern of behavior. And if you start getting out of sync, essentially,
Mhm.
you lose that oscillatory behavior.
Mhm.
Right, for example, in the example that Josh showed, the simplest kind of encapsulator where you have the three transcription factors,
Yeah.
you have to have that the pattern of expression has to be timed correctly.
Right,
And so the noise, what the noise does is it gets the timing out of sync, so to speak, and so then you lose that oscillation.
so then, but wouldn't something like that, so over age, right, would you start to see this out of synchrony start to act, like if you started in sync and then
So,
you become an old worm or something, right, and then
right,
would you start to see that loss of synchrony, would you hypothesize?
I think so, these oscillatory behaviors are studied because there's a lot of interesting, so for example, some proteins might have a certain effect if on oscillatory behavior versus a different type of phenotype if they're more kind of sustained, inflammatory response versus immunitary response, I think with the whole running signaling. So sometimes it's not necessarily that if you get out of that oscillation is bad, and it's just that literally two different phenotypes.
I see,
So I don't know if there's any example of oscillatory versus sustained expression in aging. Rear cut.
Yeah.
Okay.
Access the effective, right?
Someone. Yeah. But, it's, part of it, I think, is also, resetting it during the curriculum. Some things that always reset. Because in the previous set, from the home spaces.
Resilience. Right.
So maybe it's something like that, or maybe there's not noise per se, but induced changes in the frequency that are associated with aging or something like that, changing it up. And then if you had a robust genetic circuit to force it back in, like that could be a really powerful readout where you wouldn't even need to do sequencing or something like that, right? Especially if it's a GFP, ERP reporter type thing, right? Where you could then just literally put, in my mind words again, but put them in some sort of system where you're like looking at them for a few days or something, right? And then, but in principle for a cell culture system, I would imagine too, that could work out.
Yeah, I just struggle with 2D cell culture because anything that we study in that just doesn't correlate with in vivo.
Yeah.
From the immune side and senescence side.
Yeah, hard plastic.
Yeah, so we talked about 3D models.
Yeah.
But then you're still missing, let's say, the immune component.
Maybe like
And-
it works.
I mean, I guess I find it useful when we're trying to validate some cell-cell interaction that we found in vivo, then it seems to, I mean, you've got the two cell types very, clearly focused, but
Yeah.
I struggle with that.
Helen Wow just had I think it was published, I don't know if she gave a talk. Dialing back the stiffness matrix to physiological, which is 3 orders of magnitude. Back.
With aging, yeah.
Well, so just instead of baseline, can you get these multiple stem cells to function in vitro? And they use I think it's somewhere, it's a narrow range of function. Only from 2 and 30. It's real fun. All bits of plastic. Turns out that stem cells are ciliated. If you grow them at the wrong stiffness, they're not ciliated. And they're not ciliated on sense because of the multiple protein coupled receptors in the business. They don't function.
Wow.
Yeah.
I don't see it out yet.
Oh,
I'll look on preprints,
let's drop hell of a lot of nutrition.
With a question, so if we design a synthetic system to like improve some aging situation, like how much burden would the synthetic system put on the cell itself? Like, do we know how much burden is that?
I think it's gonna depend entirely on what synthetic system is used. I mean, even just a fluorescent reporter puts some burden on the system.
I think I mean, in that case, is it possible to design a system that only like gets turned on when the noise or certain aging related damage exceed a threshold? In that case, it's like it's off in like a normal case, and then doesn't put burden to the cell, and then it gets turned on when.
I mean, the premise of the toggle switch, if I understand it correctly, is once it's received the inducer, it stays on. However, that requires a baseline expression of the sensor. So you cannot have a system, a orthogonal system that doesn't have some sort of sensor that's constitutively expressed at some background level, then you can amplify the signal with feedback. One of the things that's getting kind of popular is integrating promoter sensing modules that can basically, you put a synthetic promoter in the genome in front of, whatever your reporter is or your system is. And so only when the transcription factors that are present in the cell endogenously are present, does it start to sense. So the DNA section is what's actually your sensor. And so it's like, quote unquote, inert. It's not having something float through the system that's toxic until the conditions are met. And these three components, for example, if you had a tripartite sensor in front of your reporter, once all three things are there, then it turns on. That's probably the closest thing to like a background free system. But that's where a lot of people are trying to do cell type specific promoters and things like that. But it's been very challenging to have it be specific to different cell types that can integrate that cell sensing system. I know there's a lot of work being done in that area. Beyond that, I don't know how to like go over a threshold and then trigger a response.
So we talked about in our last session was specific cell types and finding the cell state of specific cell.
Oh, you were
types.
with Caleb?
Yeah.
Yeah.
But actually, we wanted that first sentence.
Yeah,
Right, so
that'd be
specific
fantastic.
subtypes and then cell state information.
But yeah, that's going to be kind of tricky, because even if that's possible, like when you turn the system on, the cell could already be damaged to a point that it cannot be fixed.
It would require very precise tuning to determine that threshold set point. And that would require a lot of background context and things to be able to figure it out.
And my understanding is not even clear whether there's a threshold.
Right.
Right?
Or if it's always there.
Like then when we're born, essentially, we start aging.
Right.
And so,
Maybe that's an interesting question, is that dynamic between development and aging, when those
Yeah.
change.
We talked about that too.
talked about that.
Well, just a, recapitulation of developmental signals for tissue repair that comes with aging tissue.
Yeah. And then, like in some in animal systems, we rather some things that are lethal, if you can't make a strain of it because it's dead, but you knock it out in a tissue later on, it actually live longer. And so it's like things that you need early on in development,
Yeah.
you don't need later, in fact, it would be preferential if you don't.
But at the cellular level, there must be some threshold that says, from now on, there's resistance to apoptosis, for example, right? I understand cells kind of age and maybe you have a little bit more misfolding aggregation because of toxic stress and all that. But, there are some markers of aging, like resistance to apoptosis, like the cell or cellular senescence, right? Like resistance to apoptosis, where the cell sits there and just secretes this.
Yeah, I mean, I don't think that's a sick cell, I think that's a signaling cell. So like, but I'm a little different, I don't come from necessarily the aging field, and so I don't think it's a zombie sick cell.
Okay.
Where, resistance to apoptosis is something. It's just because it's signaling, and it's performing a very important signaling role.
I see.
So
So it's not like a switch type response, like.
There's a apoptosis?